Let's welcome Mahmoud Rahman. You're welcome, Mahmoud.
So this is forward and backward. Thank you. Morning, everyone. This is Mahmudur Rahman. I am welcoming you in my presentation on possibility of rapeseed as human food by indexing and characterization of new class of protein from rapeseed meal. Rapeseed meal is commonly known as canola, the mustard. It is also a variety of canola rapeseed. So the world is suffering from scarcity of food, especially protein. That is one of the important components of food, costliest among the components, and hard to grow, hard to obtain. The topmost cause of human death in the globe is hunger. This is more than COVID-19 cases, more than cardiovascular disease, road accident, many other diseases. About 21,000 people die of hunger every day. In 10 seconds, one child is getting death with the lack of hunger, due to hunger and malnutrition. Even in the US and Australia, the scenario is not completely good. So everywhere in the world, there are people crying for the lack of food, especially protein, including protein. And it is not an easy task to supply lots of food for world population about 8 billion people. And to back this up, to feed this huge amount of people, a continuous production and cultivation chain should be backed up. So plant-based proteins are highly thought as a potential source of protein, and they are also thought to be climate friendly. They require less amount of land area, less energy, and they are proven as associated with less emission of greenhouse gas. So worldwide, there is a big shift of consumer behavior from animal-based protein to plant-based protein. And research focus is pointed on the abundant and healthy protein source. That source is that source in my study is canola. Canola is a oilseed crop that have huge amount of oil in its seeds. After extraction of the oil, it contains about 40% protein. So it could be a great source of protein. And it is cultivated worldwide in huge amount. Australia is one of them. The Australian canola, it is mainly genetically modified, and it is a rotational crop cultivated in Australia as a rotational crop with wheat. And science has proven that this is one of the healthiest cooking oil that is widely used through the globe. After extraction of the oil in the industry, the remaining residue, that is a byproduct that contains, we call them as canola meal. It is globally produced in a huge amount. The most cultivated edible oil is soy. The residue is utilized as human food as tofu, burger, protein-based, plant-based protein burger, soy milk, soy sauce in different ways. Whereas canola meal is only utilized as livestock feed and soil fertilizer. There is no use of human food in past days. Considering the fact that these proteins are cheap, they are abundant, they have higher amino acid levels and higher protein efficiency ratio. That is 1 gram of protein. How much protein is utilized when we eat? The amount of protein utilized to form the body mass is called protein efficiency ratio. So these proteins have higher protein efficiency ratio. They used to build up our body muscle. So these facts are summarized and published as a review in the beginning of this study. In the study, we found that there was no well-developed method to analyze the protein in Brassica seeds, canola seeds. So we developed an EMS mutant population. We used an EMS mutant population developed in University of Nottingham for our analysis, and we developed and optimized a method to analyze the proteins in a single seed. We used simple SDS page analysis and Western blot, and utilizing them, the optimized method was published in Agronomy. Southern Cross University, Southern Cross Plant Science hosts the Brassica database. They have an in-house database of Brassica, rapa, seed proteins database. So we use that database to explore what proteins are inside the seed. We compared that data that is technically called BLAST. We blasted the data with known plant that is called Arabidopsis thaliana. The genetic information is well studied for Arabidopsis thaliana. We compared them and we find some candidate genes in Brassica rapa. To get confirmed about the genes, what are the proteins inside the gene, we bioinformatically did some comparison that is called multiple sequence alignment, and high degree of homology were found with the candidate genes with known plant proteins. A phylogenetic relationship was studied to reveal the genetic architecture and genome evolution, and also predict the gene functions for that proteins. 3D modeling were done for the candidate proteins, and the outcome was published in Clinical and Experimental Allergy. Traditionally, mustards and canola, they were used in diseases where bacteria are involved. They were also used in food preservation. Here also, bacteria are involved. And while we did the bioinformatics study, we find the candidate proteins have high sequence similarity and proteomic similarity with the antimicrobial peptide they were reported earlier. So we did some in silico study, and we find amino acid sequence similarity, and we did molecular docking. It also shows that the proteins are highly bind with the bacterial enzymes and inhibit their activity. In vitro analysis, we also find the proof of that antimicrobial activity. And all of these studied, all of the outcomes, they justified the use of mustards and canola seed in bacteria-borne diseases. The study was published in Frontiers in Pharmacology. As a next step, we analyzed the seed samples, the single seed. There were about 2,000 seed samples, different varieties. And we studied them, and we find that we identified the proteins in the seeds. We characterized 323 major proteins. They are abundant in seed. Earlier, it was known that albumins and globulins are the major seed storage protein. Seed storage protein are the protein that stored in the seed, and they supply nutrition when the seed germinates, and they have high potential of use as human food. So we identified a new class of plant protein that is called vicilin, and characterize the role of protein of the identified protein. To summarize these, the information could be used to determine the biophysics of protein structure, determine the 3D structure. They give us the physiological function of the protein. We can get idea about the bioactivity of the proteins. And finally, they give us the metabolic profile of the protein. So utilizing the metabolic profile, we can predict the use of the proteins as human food. Tailoring all the information, it is expected that we'll get rapeseed-based proteins in our food very soon. The study was published in PLOS One. The scenic beauty of the yellow mustard field, it will expect, it is in near future, it will not only fill our eyesight, it also it will become in our plate very soon. And more than 3,000 million metric ton of food, more than 3,000 million metric ton of healthy proteins, they will add in our food chain and help to mitigate the cry of hungry people and bring back the smile in the hungry children and population. Thank you.
So now we can welcome questions to Mahmud. Is there anybody with some questions for Mahmud? You're welcome to present them.